Today we are going to start the blast. Okay. I have uploaded all the recordings, all the PPTs in the drive. Please go through it and let me know if you have any question about it. Okay, so let's just start the blast. Can you tell me, first of all, what do you know about the blast? What is blast? Hello?
It is the basic local alignment tool that
Hmm? That?
basically that finds the region of local similarity between the sequences.
Yeah, correct. So blast, that's a basic local alignment search tool which is finding the region of local similarity between the sequences, right? It was designed to use a nucleotide or a protein sequence to search and quickly find the similar sequences in very large databases. And here, this PRAS program basically it compares the nucleotide or a protein sequences to the sequence database and calculate the statistical significance of matches, like whether it is matching or not, and then whether it is significantly matching or not. So that also you are getting by the blast. Okay, but let's talking about the blast more. Actually, Blast is developed and maintained by the NCBI. Okay, it's maintained by NCBI. Uh, then means like it is means let's consider if you are finding like if you have any novel sequence or just a new sequence, you want to find out that particular novel sequence is belonging to which species. So what you have to do that you have to match that newly discovered sequences or the sequence with all the sequences which are present in the database, right? And if it is matching, then that's a result of your sequence similarity search, and that's what exactly the BLAST is doing. It is doing the sequence similarity search to find out the relationship between the organism, to find out the functional relationship, to get the evolutionary relationship. So this blast is very important tool in the bioinformatics. Like we are saying, if you don't know anything, just do the blast. At least some information you will get there. Now, whenever you are performing a blast, you require two types of sequence. I'm not saying two sequence, I'm saying two types of sequence. So, what is the first type? That's a query sequence. Query sequence means what? It's your input sequence, which you are giving input for search. Another you are having is a reference sequence, which you are giving as a reference. That's your uh, means like you are giving for the matching for the alignment. Now whenever we are talking about the alignment, actually there are two types of alignment. First is a global alignment.
And local alignment.
Exactly, local alignment. Yes. So what is the difference between global and local? Do you know?
In global alignment, it aligns the whole sequence. It compares the whole sequence, whereas in local alignment, it compares only a segment of sequence with the query
Hmm.
sequence.
Right. So global alignment, it aligns a sequence globally, like end-to-end alignment we are saying. We are saying it's a end-to-end alignment, correct? Means it's align your entire query with the entire reference. Means this global alignment you can perform if the organisms are closely related. You cannot perform the global alignment if the organisms are distantly related, because here the length of the query and the length of the reference should be similar. And at that time only you can perform the global alignment, right? But like whenever we are getting a novel sequence, a new sequence, or any kind of sequence, not necessary that this particular sequence is going to be aligned. Uh, like it's a closely related to your reference. It's not necessary. Also, it's not necessary that the length of the query and the length of the reference will be similar, because whenever we are saying the organisms are homologous, so at that time you are saying the sequence similarity, it should be greater than or equals to 30. At that time we can say yes, the organisms are homologs of each other. So, with such kind of condition, we cannot use a global alignment if you want to do the sequence similarity search for the distantly related sequence. And that's what the BLAST is using, the local alignment. It is not using the global alignment, right? Because in a local alignment, it aligns a sequence locally. Means wherever it will find a match, it will match. Otherwise, rest of the things, it will simply leave it as a gap. Means in the local alignment, we are getting a sequences, or you can say we are getting the alignment in the form of substrings, in the form of stretches. Okay. So that's what the difference between global and local alignment. If I will show you the diagrammatical representation, you can see here in the global alignment, it is doing the end-to-end alignment. But in locals, it is just going the it is doing the alignment with a sequence similarity, like a higher sequence similarity there. Means like wherever it is matching, it will match. Otherwise, rest of the things, it is simply leave it as a gap. So likewise, we are getting in the form of stretches, in the form of substrings there. Means if you will see this difference, so basically here, like the global alignment, it's, I will just zoom it for you. Just a second. Yeah, so in the global alignment, it's like attempt made to align the entire sequence, and here it's just the region of highest level of similarity between the two sequence. Then the global alignment contains all the letters from both the query and the target sequence, but it is only aligns the substrings of the query to the substring of the target sequence. Then in the global alignment, if the two sequences have approximately the same length and are quite similar, they are suitable for global. And any two sequences that can be locally aligned as a local alignment find the stretches of sequences with a high level of matches without considering the alignment. So you can do it for the distantly related sequence also. Then it's used for comparing the homologous genes, and here used to find out the conserved pattern, domain, motifs, the two proteins. So global alignment is using the Needleman-Wunsch algorithm and it is using the Smith-Waterman algorithm. So this is what actually the difference between local and global alignment, and the BLAST is using the local alignment there. All right. Any question?
No, ma'am.
Okay. Now let's talking about the BLAST. There are three types of BLAST, or like what are the types of BLAST? Do you know?
Um, Ma'am, blast
Yes.
for blast P for protein, then blast N for nucleotide. Uh, do you mean that?
Uh, Actually, yeah, these are the types, but okay, I will tell you. What are the types of blast? So the first is the online blast. Second is a web-based blast, and third is offline blast. So the type which you are telling, sir, it's a types of online blast. Okay?
Okay, ma'am.
Yeah. Now, let's talking about the online blast. So actually the blast which you are using up to now, that's from the NCBI blast. This is the online blast. And each type of blast, like online, web-based, and standalone, they are having their own pros and cons. So if you will just go here with the online blast, you can see the home page. Yeah, this is what the homepage of the BLAST basic local alignment search tools find the region of similarity between the biological sequence. And here, if you will see that, like these are the four flavors, or you can say five flavors of the online BLAST: nucleotide, protein, BLASTX, T-BLAST-N, and T-BLASTX is also there. Now, what is the nucleotide blast? What is a protein blast? Do you know?
Ma'am, nucleotide blast is the blast in which we compare the nucleotide sequence.
To what we are compared? Yeah, we are
Nucle-
comparing. Uh-huh.
Nucleotide sequence with the query nucleotide sequence.
Reference nucleotide sequence.
Yes, ma'am.
Right. Yeah. So nucleotide blast is what? Like here, if I will show you the table properly, like here option is the program is blast n, that is a nucleotide blast. So where your query type is also the nucleotide, your database type is also the nucleotide, means you are aligning your nucleotide query with the against the nucleotide reference database, and the comparison is what? It's a nucleotide comparison, right? That's a blast n program, which means a nucleotide blast. Another type we are having for the online is a blast P, that is a protein blast, where your query is also protein, your database is also protein. Means here we are going to blast the protein against the protein database. So the comparison is what? Like here we are doing the protein comparison, right? Then the next we are having is a T blast N. T blast N, where your query is protein and you want to blast it against the nucleotide. But actually, this nucleotide, these are the translated nucleotide. If you will see here, T blast N, like a protein against the translated nucleotide, right? So that's what the comparison here is, again the protein comparison. Another we are having, that is a blast X, where your query is nucleotide and you want to blast it against the protein database. But here, the nucleotide is also the translated nucleotide. So that's why the comparison is here protein. And another we are having is T blast X, which is like a translated nucleotide against the translated nucleotide, and that's why the comparison is here also protein protein comparison. So these are the five flavors of the online blast. Is it clear?
Ma'am, the translated nucleotide is mRNA, right?
No, it's a protein translated. It's not transcribed. Transcription, no.
Okay, okay, ma'am, got it.
Yeah, yeah. Okay. So, regarding to this table, we have some questions. Will just theoretical questions. I will ask you, and you can tell me answer like which type of blast you have to use at that time. So, just read the question. You have a protein sequence, and you wish to know what other proteins look like. You have a protein sequence. You wish to know what other proteins look like. Which of the five basic BLAST programs should you use? Which program you will use?
Blast P.
Okay, what about others?
Ma'am, same, ma'am, blast P.
Okay. Mm, what about others? Is there any another answer? Yeah, Namrita ma'am, Sonali ma'am. Yeah,
Ma'am, can it be blast and T blast and
okay. Okay, so whoever is saying like blast P, uh, okay, first of all, Annapurna ma'am, can you tell me why T blast N?
ma'am, I have just predicted that if we have a protein sequence and we have an unknown protein, so
Uh-huh.
the translated nucleotide can also be an option.
But why we need for the translated nucleotide? Because if it's a translated nucleotide, it is coming for the DNA search, right? So it's a blast P because it's very simple that if it's a protein, you want to find out other proteins. So comparison is protein protein only means your query is protein, your target is protein, and your comparison is also the protein protein. So obviously it's a blast P. Are you getting my point?
Yes, ma'am. Just for curiosity, I have said.
Yeah, yeah, no problem. It's okay. But now it's clear why blast P.
Yes, ma'am.
Okay. Then next question. You have a DNA sequence and you wish to search for other DNA sequence to find one that encodes for same or similar protein. Which blast you will use? Read the question very carefully. What is your query? What is your reference? And what is the comparison?
Ma'am, here I think so it will be blast X.
Okay. What about others?
I feel it's T Blast X.
Okay. Uh, Blastex. Who is saying the Blastex?
Ma'am, it is blast X, I think, cause there
Okay.
what was the answer we want to compare for similar protein.
Hmm.
The like our input is which DNA is encodes, like
Hmm.
sequence which encodes. So I think so there will come translated nucleotide, and the other one will be protein. So I think it's blast X.
Okay. And who said the T Blastex?
I did.
Okay. Can you tell me why?
Yeah, because our query is also nucleotide, and we are searching for other DNA sequences,
Hmm.
and we want a protein or
Hmm. Yes,
comparison. So that's why.
correct. Yes. So actually, the ISR, the thing is like here you can see we want a DNA sequence, right? We want to search the other DNA sequence. So our query is what? DNA means nucleotide. Our target is nucleotide. But we want to find out the protein. Means what? A protein comparison we want to do. So in that case, if you will see if your query is nucleotide, reference is nucleotide, but you want to find out other proteins. So at that time, it's a protein-protein comparison. That's a T-blast X. Is it clear?
Okay. Yeah.
Hmm. The third one, you have DNA and you wish to find other DNA that's look like which you will use. Just wait, Ayesir and Sonali ma'am. What about others like Sana ma'am, Namrata ma'am, Tanya ma'am, Annapurna ma'am? Read the question carefully.
Ma'am, blast and
Yes, right. Blast and so the answer is same from all.
Yes, ma'am.
Yeah.
Yes, ma'am.
Hmm. You have a DNA and you wish to find other DNA. That's yeah, that's a blast and program. Now, the last question, you have a protein sequence. You wish to search the DNA database to find the gene that encodes for similar protein. So, which you will use?
T blast N.
Yeah, T blast N.
Yes,
T blast.
correct, correct. Yes. So, that's what like your queries protein, your references means your target is nucleotide, and comparison is protein protein. So, it's T blast N. Yes, exactly. So this is what like first of all we need to be understand because this thing should be very clear that which type of program we have to use then and then only we can get a proper result, right? So I hope it is clear to you everyone. It is clear to everyone that what program we have to use and what is the meaning of it. Correct?
Yes, ma'am.
Now let's go with the nucleotide blast. Just see one example for the online blast. So here you can see like five programs we are having. So which type of blast you want to perform, accordingly you have to select the program. Then here you are having the search box where you can give the accession number, you can give the gene identifiers, you can give the FASTA sequence also. Then, if you have a FASTA file, you can just choose your file. Or if you want to align more than two sequence, you can just go with the align two or more options there. Okay. Then, once we have given the sequence, next it is asking to search the database. So, I will just take one example of PRNP gene sequence, just a small stretch of PRNP gene I am going to take. Or maybe any another gene. Hmm. I'm just going to take one very small stretch of sequence and then I'm going to paste it. Okay. So once you have pasted, if you want to keep the job title, that is completely the optional part. So you can give. Then here it is asking the database because you are going to be align your query against which database. You have to select the database for that. Like standard databases in that you are having the options like nucleotide, RefSeq, whole genome, targeted loci, and many more. Uh, So for that, from that, I'm going to select the nucleotide collection for now. There are some another databases also you can see, but if you will see that like we are having very limited options over here, right? Out of that only we are selecting. We cannot give the database as our choice. Like we cannot add there. We cannot remove anything there. So that's a limitation you can say. So I'm just selecting from now the entire nucleotide collection. Against that, I want to align the query. Then the organism. So organism, it is asking actually, uh, like if you want to align your query against the specific organism, let us consider I want to align this query against the Homo sapien only. I can give the Homo sapien, so it will show you the query against the Homo sapien reference only. That is the one thing. But maybe another thing it can be like you want to align your query against all the organism except Homo sapien. So at that time, you can also give the Homo sapien over here and then you can click on exclude. So it is going to be exclude that organism. Okay. Then here this is what like limit option like entrace query if you have then we want the higher similarity searches only, and then you have to click, you can click on the nucleotide sequence using the mega blast and then click on the blast. What happened? Just a second. Yeah, okay. Yes, it generated the result. Maybe I will share the query in the chat box. Maybe if you want, you can just try from your side also, like a small stretch of query I'm taking. And here you can see that you are getting the result. So what you are getting, first of all, the job title. What is the job title of your entry? Then reference ID here. Then you are getting the program. What program we use? Blast and nucleotide blast. What database we use? It's a nucleotide database. Query ID. Then here you are having the description for your query. Molecular type, it's a DNA, and the query length, how much query length we have taken. Then if you will go down here, so you are having the description. So in the description column, you are getting the description of alignment, like your query is aligned to what reference. So that description of the alignment you are getting over here. Then you are getting the scientific name of the alignment. Okay. Sorry, scientific name of the organism, to which organism your query is aligned. Then here you're getting the max score, that is a maximum score of the alignment and the total score of the alignment. But out of that, all columns, the query coverage, e-value, percentage identity, these three columns are very important. I'm repeating again, query coverage, e-value, and the percentage identity, these three columns are very important. And out of these three, we have to first focus on the percentage query coverage. Okay. So do you know, first of all, what is the percentage query coverage?
How much percent the our sequence is aligned with the reference sequence?
Uh, Means like a base wise alignment you are saying, like how the base from a query is similar to the
Yes, yes,
reference.
yes.
Okay, but then what is the percentage identity then?
Is it with the organism?
No, no. Actually, percentage query coverage means what? Like how much your query is covered by the reference. Okay, I will just write over here percentage query coverage is nothing but like how much query covered by the reference. That is what a percentage query coverage means. Let us consider like this is what your query. Okay, sorry, this is what your reference and this is what your query. Okay, like this is the reference, this is the query. So here the reference is only able to cover your half of the query. Okay, the reference is only able to cover your half of the query. So at that time you can say percentage query coverage is only the 50%, like your 50% query is covered by the reference. But as of now, if you will see, only half of the query is covered. So you are missing the information of your half of the query, and we don't want to miss any information. That is why percentage query coverage should be high, right? Means right now, if you will see the example here, let us consider this is your reference, this is your query. Now your query is fully covered under the reference. So at that time, we are saying the percentage query coverage is 100% because your query is fully covered by the reference. I'm not saying like that the percentage query coverage should be always 100, but higher the query coverage, better the alignment, you can say. Is it clear, first of all, the percentage query coverage?
Yes, ma'am.
Yeah, Sonali ma'am, is it clear?
Yeah, yes, ma'am.
Hmm. Now, from the query, how the basis from the query is exactly similar to the reference, that is what the percentage identity. Okay, percentage identity, it is showing you how the base from the query is exactly identical to the reference. That is what the percentage identity, like a base wise it is checking. But now I have told you that like out of the query coverage and percentage identity, you need to be first focus on the percentage query coverage and then on the identity. So why it is like that? Any guess? Why percentage query coverage is important than the identity?
Ma'am, because if it's only 50% covered, then it doesn't
Hmm.
matter that how much percentage identity is there. We have any,
Yes,
our whole sequence is like covered. That's why our whole query
exactly.
is covered.
Correct. Very correct. Means if you will see that here, like the previous example which we are taking, uh, so here the percentage query coverage is 50%. And in that 50% query coverage, if the all bases from the query is exactly aligning to the reference, your percentage identity will be 100% high. Correct? But you are missing your 50% query information. So at that time, there will be no use, even if it is showing the highest percentage identity. That's why first the query coverage, then the percentage identity. Even if you want to filter out based on the alignment, first give the preference to the query coverage, then to the identity. And here, if you will see, like Some of the examples, percentage query coverage is 65, but the identity is 100%. So at that time, can we say it's a good alignment?
No, ma'am.
No, correct. So percentage identity, percentage identity is nothing but the uh, base from query is aligned to reference. How much? Base, basis you can see aligned from the query is aligned to the reference. That is for the percentage identity. Next we are having is the e-value. Do you know what is the e-value? Any guess what is e-value?
Ma'am, it's
Error?
the expected value.
Yeah, right. And what it is giving? So e-value that stands for expected value. Actually, this e-value it will give you the number of hits, or you can say the number of alignments which occurred by chance. Okay, you are getting the number of alignments which occurred by chance. Means this e-value actually it will tell you the significance of the alignment, that whether your matches occur surely or whether your matches occur randomly. So if the randomness of the alignment is increasing, the e-value will also increase. So do you want the random alignment? We don't want the random alignment. We want a sure alignment, like a fixed, like we want a sure alignment, right? So that is why lesser the e-value, better the alignment. Because we don't want those hits or those alignment which occurred by chance, correct? Are you getting my point?
Yes, ma'am.
Yeah, ma'am.
Is it clear, or I can repeat it once maybe? So this e-value actually it is telling you the significance of the alignment. Okay, so I will just keep. So lesser the e-value, better is the match. Best match is like e-value should be 0.0. Best match you can say at the e-value 0.0. Have you ever heard about the p-value? Something? Okay. Now, like this is what here if you will see like in the offline, sorry, online blast, you're having the graphical representation also summary, which is showing you the graphical representation based on the alignment score. Like if alignment score is greater than 200, red, between 80 to 200, purple, then 50 to 80, green. Likewise, you're getting the alignment visualization. Here you are getting the base-wise alignment also, like what is in your query and what is your subject. Subject is nothing but the reference. So base-wise alignment also you are getting over here. So this is what general, the overall idea about the online blast. So right now, if I will tell you what are the pros and cons of the online blast. So online blast actually it will give you the quick time solution. Like if you have query, you want to blast it against the protein. Sorry, if you against the reference, so uh, you can go with the online blast. Then it has a good graphical representation. Results are very user friendly. It's very easy to understand by the graphical view and all. Then, as it's an online blast, so obviously without the internet connection, we cannot run the online blast. That's a very obvious thing. Then next is like here, uh, you like the query size is very limited. Means if you have a thousands of queries sequence, you want to align that thousands of query sequence in one go in of online blast. It's not possible. You cannot align a thousands of queries together. Or maybe if you have a query size, maybe with a 20 GB file size. So at that time also, it is not possible to align your query against the in the online blast. There is a size limitation. Okay, query size limitation there. And next is what? Like here, database customization is not available. So what is the meaning of database customization? Like here, during the online blast, we need to select the database from the list which they have provided. And all those databases are online databases, right? So if you want to add some sequences there, you want to remove some sequences there, so you cannot do like that. Means like let's consider you have some sequence. You want to align that sequence against the few human sequences, few chimpanzee, few mouse sequence, few rabbit sequence. So you cannot make your database according to it, which means like you cannot customize the database there. Okay,
I didn't
are you getting my?
understand.
Huh?
I didn't understand this part.
Okay. Yeah, no problem. I will explain again. So, here, whenever you are checking in the online blast, like we have this database, right? And I'm selecting the database from this list, correct? But now in this database, let us consider from the nucleotide collection only. I want to align my query against only the few sequence of human, few sequence of mouse, few sequence of rabbit. So can I make the database like this? Can I do the customization here?
No.
Can you add the sequence? Can you delete the sequence from here? No, right?
Yeah.
Yes, that's what the drawback. The database customization is not available over here. Okay, got
Yeah.
it?
Okay. Yeah.
Hmm,
Uh, yeah, ma'am, got it.
hmm, hmm. So yeah, this one also left actually the question. This DNA is from a real cDNA. There are no sequencing errors in it. You have to search the DNA database of BLAST looking for other DNA molecule that encodes for similar protein, which type of BLAST program you should use.
It's the T plus X again, no?
Yeah, sorry. What? Can you just repeat once, please?
Yeah, is it the T blast
Just
X?
a second. T plus X, you are saying?
Yeah.
Okay, what about others?
Yeah, ma'am, that only T blast X.
Correct, T plus X it is. I want to give one question right now to solve. Just a second. Hmm. So, visit NCBI site, perform blast P for SOX15 protein sequence for human. What you have to do is you have to just do the blast P for SOX15 protein for human. You have to download the SOX15 protein. You can download it from NCBI or Uniprot wherever you want and perform the blast P. Can you try? It's done. Would you be able to perform it?
I'm getting a error.
Yeah. What error? Tell me.
Subject string not found in CGI context.
Yeah, actually, sometimes we are getting the error. Just reset the page and do it again. I'm facing the same problem.
Okay.
Yeah.
I'm done, ma'am.
Okay. So, okay. One more question I will give if you will be able to perform, and you have to decide now which database and all you have to use there. Okay. Just a second. Or at least explain me what we have to do in that case. Means like you can tell me so that. So I'm giving one like sequence to you. So what you have to do is like just let me see if I have that question. Yeah. I'm sharing my screen. Just a second. Yeah. In the PPT only itself we have the question. Like if you will see, we have the questions like report the organism of given sequence. What you have to do is like just whenever you are pasting the sequence, you just have to give the enter once. First line should be the, I will show you how to do it. Like first line should be the greater than symbol only because it will show you the error. And like once you will give the enter and then you have to paste it. Otherwise, paste it and then give the enter. Are you getting my point? First line, it should be only have the greater than symbol, and then second line, from the second line, your sequence should be start. Okay, clear?
Okay, ma'am.
Yeah. So what you have to do is like I will share this PPT. You have to report the organism of given sequence. Okay, so I'm sharing the sequence to you right now, and you have to tell me which organism sequence is this. Yeah.
Miss, it's Candidatus nanoacylus acido bovine.
Yes. Yes. Correct. So, is it clear to everyone? Like, what you have to do is you have to paste that sequence in the nucleotide blast and again align against the nucleotide collection. Okay. Now, if I will ask for this next question, report the presence of 3D structure of given sequence and visualize it. So, this is what a protein sequence. You can see it's amino acid sequence. So how can we report the 3D structures? Like I want to know for this sequence, the 3D structures are present or not using the blast. So how can we do that? Maybe can you just tell me the procedure if you know? Or I will share the sequence. You can have to try. You can try it. The question is clear. Are you getting my point? Like, you are you able to perform it? I will give you a hint. You have to do the protein blast P against the PDB database because we want to find out the 3D structures. So the database we have to select as a reference database as a PDB database. Okay.
Yes, ma'am.
Hmm. So this is about the online blast. We'll see about the standalone blast, means like offline blast, from tomorrow. We are going to perform a practical question for it. So yeah, and we have first of all we are going to install it and then we are performing the practical question. Okay.
Okay, ma'am.
Okay. So all right then, that's it for today. We'll discuss rest of the part by tomorrow.
Thank you, ma'am.
Yeah, you're welcome. Bye everyone.